All right, awesome. So yes, we're very lucky to have Professor, or soon, very soon to be Professor Marco Messina here with us. We actually tried to get him here to Northeastern, but I guess Delaware took him away. But that's okay. We actually overlapped when we were graduate students at UCLA, and he worked with Heather Maynard and Alex Spiconi there, and then moved up to Berkeley, where UC Berkeley, where he's been basically a postdoc since, working with Chris Cheng, if I'm not mistaken.
Yeah,
and now is starting his faculty position, independent position now at University of Delaware very shortly. So, thanks for being with us, Marco, and please take it away.
Yeah, sure. I guess I will share the screen now and do this thing and then swap, and it should be the right one. Yeah, so thank you, Stephen. Thanks for inviting me and having me. I always love doing these types of things, even though it's quite hectic now. I found out yesterday my wife came down with COVID, and so I've been scrambling to like complete all experiments and finish presentations and deal with undergrads here. So I apologize for the tardiness, but yeah. So I'll talk about my trajectory for a bit. Everything's kind of jumbled together, so I'll incorporate my trajectory, some research that I did along the way, and then I'll sprinkle in little bits of ways in which I have used computations or have collaborated with people that do computations towards my own research. I personally don't do computations beyond like a ChemDraw level of theory, but definitely I appreciate the things people like Stephen do. I just leave it up to the experts. I don't try to do that myself. And so this is kind of the broad trajectory into science that I've had, somewhat non-traditional in a way. And so I started out as most people do as a child. And so really, I didn't really care much about science, or I wasn't really a good student in high school or in undergrad for that matter. And so I really just wanted to play soccer and be a soccer coach. And that's what I was going to college for, be a high school teacher and do that. And so I went to Texas A&M University in Corpus Christi, Texas. So that's a primarily Hispanic serving institution. And what happened there was somewhere along the way, maybe in my third year, things kind of shifted, and I met a really young, passionate professor in my organic chemistry course that got me into his research group and really changed my trajectory. And so it wasn't until my third year that I got into research that I started really studying and taking science classes. And I quickly started becoming involved in programs with the goal of recruiting and retaining underrepresented minorities in science, like the Lewis Stokes Alliance for Minority Participation, SACNIS as well, and really finding and building on a community through those initiatives. And I'm still somewhat involved in these and also others. And so that propelled me to go get a PhD in chemistry at UCLA, where again, I was involved in various organizations with similar goals. And now I'm a UC President's Postdoc Fellow here at Berkeley with Chris Cheng, as Stephen mentioned, and also through the Mosaic K99R00 program. And so, I also forgot to mention that people should feel free to chime in and ask questions along the way or something, to make it a discussion as well, if you like. I certainly won't mind. So, as I had mentioned, really my whole trajectory changed when I met this one professor, and I started taking organic chemistry. And organic chemistry really interested me because that's, to me, that is when chemistry started making sense. Learning how to build molecules, how to make molecules, visualizing these molecules on paper was a really big deal to me, because in Gen Chem, you're really just doing all this math and everything is so abstract and it's really difficult to understand. Whereas in organic chemistry, you're really drawing things out, you're drawing orbitals, you're learning all these little rules. To me, it was a lot like playing Legos, which I did quite a bit of as a child. And so that really drew me. And Mark Olson here, Professor Mark Olson, who's still at Texas A&M Corpus Christi now, got me into his lab and really heavily mentored me. And so this is a lot of the stuff we did in that lab. And so I really started out as a synthetic organic chemistry making molecules. And so I made these really extravagant large molecules. What they are really, doesn't, what they're called really doesn't matter. But what we were studying here was what's called host-guest chemistry. And so we have these really electron-poor, electron-deficient molecules out here, these horseshoe-shaped, I should get the laser pointer, these horseshoe-shaped blue boxes, which are electron-deficient. And then we have these electron-rich molecules here denoted in the yellow. And so we can modulate this self-assembly process. So they self-assemble just like this upon treatment with sodium hydroxide, and we can turn this off with hydrochloric acid, so HCl. And so we can modulate this switching via acid and bases, study how strong this non-covalent interaction is, and then we can visualize this. And so essentially what we're forming here are micelles or bubbles, and we can modulate the formation of these micelles through either acids or bases. And so this is actually a crystal structure. So this is the actual atoms and how they're aligned in the solid state, which is quite interesting. Again, it's all visual, and that's what really got me into it. And so with his support, I was kind of going through a phase where I really thought I wanted to be a doctor. And so I was pre-med, probably like many other undergraduate chemists. And so what I did was I applied to a bunch of different summer research programs. Half of them were these clinically based programs at medical schools to do rotations, and the other half were research-driven, university type settings. And so I didn't get in any of the medical school ones, and I did get into some of the research school ones. And so I ended up doing summer research as an undergraduate right across the Charles River at MIT, where I worked with this guy named Jeremiah Johnson, who was also starting out, brand new guy. And this is where I got my first whiff of polymer chemistry. And so what we were doing here, where we were developing new ways to make cyclic polymers, which is very difficult to do, but really interesting because cyclic polymers contain many different interesting properties over linear type polymers. And when you think of linear polymers, you're pretty much surrounded by them. Plastics are linear polymers, your clothing, if it's synthetic, it's probably a linear polymer, the tires on your car, the chairs you're sitting on, all linear types of polymers, different types of polymers. And after that, I came into UCLA. And so I'll talk about some of this research as well as some of my postdoc research after this, where I worked with two people, Heather Maynard and Alex Spiconi, so two bosses. And I got to do a whole lot of different things, but still in the realm of synthetic chemistry. I was still making things, which is what I enjoyed to do, what I enjoyed doing. And so my chemistry really spanned a whole variety of fields, from polymer chemistry, where we were making new methods to develop polymers using visible light. So we can have these cluster-based species, these boron clusters, mixed in with some sort of monomer that we're interested in polymerizing, shining a light, and then getting a polymer, which is quite interesting to us. We also had these different types of research themes where we were making glycopolymers, so polymers based on sugars, and we use these polymers to stabilize protein therapeutics. So protein therapeutics is a really large, really has a large pharmaceutical market share. It's over $20 billion with in the market right now of protein therapeutics, but they suffer from this problem in that they're very difficult to store. Proteins, one, don't like to be outside of the body, and they degrade really quickly under certain conditions. And so to alleviate that, many pharma companies put in excipients, or they add in things like amino acids, they add in sugars to stop that degradation process. And so we were investigating new types of materials to do that. And then I also had various interests in using metal-based chemistry, so organometallic chemistry, in order to chemoselectively modify proteins. And so I'll kind of talk about that a bit more, and then I won't really talk about this, but I did have a large hand in developing atomically precise cluster molecules. So I'll kind of talk about the gold-based arylation work, or using organometallic reagents to modify biomolecules. And so the chemoselective modification of proteins and protein therapeutics like antibodies is extremely important. So antibodies are extremely large types of proteins. They contain they're over 80 to 140 kilodaltons in size, which means they contain well over hundreds of amino acids. So it's really difficult to modify just one type of amino acid over all the other hundreds that might be present on the antibody. So a lot of effort goes into identifying chemistry that will enable you to selectively modify one amino acid over this whole array of others. And so there are many reasons why you might want to modify antibodies. And so I put the biggest reasons here on the left. You might want to attach a drug to it. That's commonly done. There exists many types of antibody drug conjugates on the market today, because antibodies are very good at targeting specific sites on cell surfaces, or specific proteins on cell surfaces. And then when you attach an active drug to them, then that's a way to target that drug selectively into specific types of cells that express specific types of proteins. There are also, you might also want to study protein localization, where protein is within a cell or in the body. And that way you would attach some sort of dye. And so there are many fluorophore protein conjugates, because fluorescent dyes give you this nice bright signal and enable you to see specific proteins in a cell. And then there are also many different types of polymer protein conjugates available on the market today. I think there are like 12 FDA approved protein polymer conjugates available, because PEG increases the in vivo lifetime of the protein therapeutics. So proteins degrade rapidly inside your body. You can't just swallow a protein and expect it to do anything. But PEG enables this protein to be much longer lived. And so there are different kinds of ways to modify proteins that I put here that I won't get into. There's malimide conjugation, there's oxidation and reduction, but many of these suffer from certain pitfalls like selectivity. And so what we were most interested in, this process termed cysteine arylation, which is forming a covalent bond between sulfur and a carbon and an sp2 hybridized carbon, like one you would find on an aryl ring, right? And we were interested in doing this using organometallic reagents. And so what I'll kind of show by this slide here is that traditionally organometallic reagents have been viewed as extremely sensitive to the types of reactions that would promote biomolecule modification. So typically and historically, organometallic reagents have been known to degrade in the presence of oxygen and water, which are exactly the types of conditions you're going to perform this type of chemistry in. And it wasn't until 2015 where a paper came out where this group used palladium-2 oxidative addition complexes to actually promote this chemoselective modification of cysteine amino acid residues on proteins and antibodies in aqueous solutions. And this was actually highly efficient. So they were forming these aryl cis bonds on whole proteins using palladium, which had historically been viewed as really sensitive to these types of conditions. And this actually worked quite efficiently. And so our group came in and we decided to explore this chemistry more and use different types of metals. And we hypothesized that gold would be a very good next metal to study. And you might be wondering, why gold? Why choose the most expensive metal, probably one of the most expensive metals in the periodic table? But gold has some very favorable properties. So gold in its +3 oxidation state is very electrophilic and it's very thiophilic. And so we hypothesized that this high thiophilicity of gold would really enhance the selectivity towards cysteine amino acid residues. And because through the course of this process, we presumably generate these byproducts that are gold 1 oxidation state. And because gold 1 is very difficult to oxidize back up into its +3 oxidation state, we thought this byproduct would be very benign. And so we have a couple of things working for us. Gold is very thiophilic, which should promote its selectivity towards cysteine amino acid residues. The byproducts generated should not be reactive because it's very difficult to oxidize gold 1. And also, the byproducts should hopefully be relatively non-toxic. So gold, people eat gold leaf. It's quite non-toxic, right? And those are all the things I basically just said. And so again, we took these commercially available gold 1 complexes that are stabilized by this methyl dalphos ligand framework, and treatment with aryl halides in the presence of the silver hexafluoride timonate under these conditions produce these gold 3 oxidative addition complexes. And so this is a very fast reaction. It happens within a minute, and notably, even though the first time I ran this reaction, I did it in a glove box with the rigorous exclusion of both air and water, which are traditionally viewed as bad for metals. When I performed this reaction outside in the hood with squirt bottle solvents on my bench, it still worked extremely well. And so I still got an instant color change upon addition of these reagents, and I still was able to obtain these gold 3 oxidative addition complexes in very good yield. And I was able to make all these different varieties of gold complexes, all harboring these different aryl groups with different substituents. And they maintained high stability when stored outside as well. And so we can obtain solid state crystal structures of all these complexes. And so again, these solid state structures are the precise location of all these different atoms in space. But what we were most interested in are not just making gold complexes, right? We're interested in seeing, will these promote the chemoselective modification of one amino acid residue over many others in conditions that are amenable to proteins, right? Water and air. And so I performed this experiment. And so I took a solution of glutathione. So glutathione is a really small peptide. It's composed of 3 amino acid residues, and it contains 1 cysteine amino acid. So it's a good model because it's cheap, and it just has 3 amino acids. And so I take this solution of glutathione and buffer, water, acetonitrile. I treat with this gold reagent, and pretty much in quantitative yield, I'm able to transfer this aryl group onto the cysteine amino acid residue of glutathione. And so what you're seeing here is a LCMS trace, so it's a mass spec trace with a HPLC, which separates out all these different compounds and gives me a mass. And so I know from the mass spec that this corresponds to this product, and I essentially have no leftover glutathione, which means it's quantitative. And so what was really cool about this chemistry is that not only is it working in water, in buffer, it also works across a wide pH range. And so we can treat in 10% TFA to get essentially quantitative conversion of this modified glutathione all the way up to sodium hydroxide, and we still observe the modified peptide. So this gave us indication that this one was a very fast process and really tolerant of a wide array of conditions. So this made us go to even other conditions and have some fun with it. And so really to push the boundaries of this, we performed this reaction in more unconventional types of solvents. And so we took this now CF3 modified gold 3 reagent, again in the presence of glutathione, and this works extremely well even in Lone Star beer, Jim Beam honey, and Diet Coke. And the cool thing about this experiment is on the LCMS, you can readily see, one, the amount of caffeine in the Diet Coke, but also the amount of aspartame. So aspartame is the sugar, right? So it's really efficient chemistry. But we could do more than just add in small aryl groups onto short peptides or short tripeptides, right? We can make a gold derivative that has this type of molecule. So this is an anti-cancer drug, trametinib, and we can transfer this anti-cancer drug chemoselectively onto the cysteine amino acid residue of this longer peptide, and we can do it very efficiently. We can also make protein polymer conjugates. So this is quite important as well, because we're showing that this process is selective for cysteine amino acid residues in this really large protein, right? So this is what, an 17 kilodalton protein that we have here, and we're only modifying 1 amino acid residue, and we're modifying it with these somewhat long oligomers. So we're modifying a really large protein with the long oligomer. So it's really efficient chemistry that's traditionally viewed as quite difficult to do. And so in under 30 minutes, we can see quantitative yield of both monopegylation, so 1 polymer, and dipegylation, as this protein contains 2 cysteine amino acid residues. And so this protein also is fibroblast growth factor 2, and it's used in wound healing process and wound regeneration. and it's also quite sensitive as well. So pegylation is pretty important. And so, you know, I obviously been away from this chemistry now for about 3, almost 4 years, but there's a lot of work being done currently in modifying the ligand framework around these organometallic reagents. I mean, and that's a big part of computational chemistry as well, because we constantly need new ligand scaffolds. You know, I didn't mention this, but I was working on this project for 8 months before it actually worked, before I got one experiment to work. And that was because I was scoping out different ligand scaffolds before I came across this one. And so now a big push is, one, to make this a catalytic process. So what we're doing here is we're just transferring one equivalent of this polymer onto a cysteine amino acid residue. But what if we can make it catalytic such that we can essentially throw in just one equivalent of metal and just have it turn over and churn out more product, right? And so that's kind of a big push now. But what I want to get to is my postdoctoral work with Chris Cheng. And it's kind of a long story why I decided to do a postdoc with Chris, but you know, the short story is a lot of the you know, data that you saw here, right, is just mass spec data that really is just, you know, a line on a spectrum, right? Or mass spec data that looks like this. And so I really had this background in synthetic chemistry, in making molecules. So one, I wanted to get into more biologically inclined science. I still wanted to make molecules, but I wanted to transfer them directly into more complex systems. And so, and I was kind of tired of looking at data that looked like this. I kept seeing all these papers published by Chris and various other people in chemical biology fields that had these really pretty pictures in their papers of cells and zebrafish models that were lighting up, and they were making these molecules and you know, throwing them into cells, throwing them into in vivo mouse models, and showing that on a microscope. And I wanted to be able to generate that type of data and to really carry over the stuff I was making into these complex systems. That's not to say that the stuff before was not complex, it was just a different type. We were just trying to solve different types of problems, right? And so really what I'm doing in Chris's group is I'm now still making molecules, but I'm making different types of molecules. I'm making fluorescent probes. And here I'm making fluorophores that are non-fluorescent in their closed state, but when they sense one type of reactive oxygen species, like hydrogen peroxide, they uncage and become fluorescent, right? So they only become fluorescent when they undergo a reaction with one type of reactive oxygen species. So why is this important? Well, we're interested ultimately in studying reactive oxygen species in cellular context. So it's kind of, it presents us a dichotomy, because reactive oxygen species have historically been viewed as detrimental to our health. But we now have quite a bit more nuanced view of them, and we know that they are important in cellular signaling processes. And so our body produces various reactive oxygen species from oxygen, for example, in the mitochondria. The mitochondria is a really large producer of these ROS species just through normal respiration processes. And all these ROS are highly reactive. They can interconvert with each other, and they're really short-lived. So it's really difficult to capture these species because they're so short-lived, so reactive, and they exist in really low concentrations. It's really difficult to track them. And the problem is whenever they're misregulated, whenever their production is misregulated, and your body produces, or your cells produce too much of these, this leads to various pathologies. So it's a hallmark. Increased ROS is a hallmark in some types of neurodegeneration, cancers, and various autoimmune disorders. And so it's really important to study these. And so there exists many different types of proteins in the cell, along the cell membrane, and in various organelles in the cell, such as NADPH oxidases, which generate hydrogen peroxide. And so they're able to take oxygen, convert it to superoxide, or one enzyme converts it to superoxide, and then NADPH oxidase convert that superoxide to hydrogen peroxide. And H2O2 is then considered a reactive signal that can go on to communicate and regulate many different cellular processes. And so that's what we're interested in studying. We're not interested only in studying the misregulation of peroxide. We're also interested in tracking its role in various cellular signaling processes. And so I kind of got at one of the difficulties, which is it's really difficult, one, to study this really small species in this pool of all these other species that look similar, right? These all look quite similar to each other in structure. They're all very small, they're all short-lived. But to selectively do that in a cell, when you have all these other structures, the challenge becomes much more, because, you know, in undergrad and probably the first half of grad school, I always thought of a cell like this, right, as this kind of empty thing with small structures inside of it. But really, it's highly complex and there's very little free space. So cells contain all these different organelles that all function differently, that all have specific functions all throughout the cell. And so this is actually a 3D microscope image, a cryo microscope image showing a cell. And you can see how crowded everything is. So not only are we trying to track these really small molecules that weigh, 18 Daltons and are highly reactive, we're trying to do that selectively in all of this. And so the traditional way that molecular biologists analyze something in a cell is through binding-based approaches of a specific analyte. And so they designed tools that would fluoresce upon binding of some sort of analyte that they were interested in. But our group makes use of a different approach. Our group makes use of a reactivity-based approach to this, where the uncaging of the fluorophore is mediated through the reaction with a specific analyte. And so through the years, our group has made an array of molecules to detect all sorts of different small molecule species that are relevant in biology. So carbon monoxide is relevant, formaldehyde is relevant in one carbon metabolism, hydrogen sulfide, and I've been working on hydrogen peroxide in particular. And so we make use of just simple organic chemistry concepts. This is a simple deprotection reaction of an aeroboronate ester. So this undergoes selective deprotection in the presence of hydrogen peroxide. So you get nucleophilic attack of this oxygen atom on the empty p orbital on boron. You get this rearrangement process, and then you get hydrolysis to produce this phenolate or phenoxide that's uncaged. And when you put this type of scaffold in a fluorophore, like this fluorescein analog, Tokyo green analog, you get this high, really bright fluorescence response when it's uncaged. And so because we're making use of reaction-based chemistry, this is a highly selective process. So it's very selective for hydrogen peroxide, even in the presence of other reactive oxygen species and reactive nitrogen species that are present in biology. And so I'll make note here that there exists a really large need for us right now to try and develop new types of triggers, new types of selective triggers. And I would argue that, you know, computation, we do run some computations to try and figure things like this out in the lab. I don't think we do it as well as a computationally focused lab would do. And I think definitely molecular biology or chemical biology really needs some help in this case. And we need help identifying new selective triggers for not only hydrogen peroxide, but superoxide. And nitric oxide is very important. And we also need help in terms of figuring out triggers that are much quicker than this. So the kinetics of this uncaging process are relatively slow. And there's quite recent work that's being done on developing much quicker, much faster triggers, which I'll get to a little later as well. And so our group has made use of this type of fluorophore scaffold to identify a bunch of different things within biology. We've identified specific types of proteins that are able to uptake hydrogen peroxide. And this hydrogen peroxide then mediates different signaling pathways intracellularly. We found that hydrogen peroxide is involved in neural stem cell proliferation, so neurogenesis, the creation of neural stem cells. And also that there exists significant crosstalk between hydrogen peroxide and other reactive species like hydrogen sulfide. And so we've been able to identify all this just using our chemical tools. But what I'm trying to study now is not signaling processes inside of the cell. I'm interested in studying transcellular signaling processes and studying signaling from one cell type to another cell type that are mediated by these highly reactive species. And so to do this, we have to overcome a limitation of traditional probes. Our probes are really good at identifying selectively peroxide and giving us a really bright signal. But the problem with these is that they are not trapped inside of the cell. They can easily leak right out. And that really hurts us in terms of signal. So we lose a lot of signal in a relatively short time, and we don't get any spatial resolution. So what does that mean? So that means that once this uncages and we get a signal, the signal diffuses all around the cell. And so we don't get information about where these increases in hydrogen peroxide are seen within a cell, next to organelles and things like this. We lose all sorts of information. And so to overcome that, we've been designing new types of systems that are cell trappable. And so here we make use of what's called orthoquinone methide chemistry. So we install a proximal fluoromethylene unit here. And so in the presence of peroxide, we get this really selective uncaging. After fluoride elimination, we generate this orthoquinone methide intermediate that's highly reactive and can go on to label nucleophilic residues on surrounding biomolecules within a cell. And so because now they're labeled on a protein, they're trapped intracellularly. They're not going to leak out. And so this really enhances our spatial resolution because we're getting more information about where peroxide is being generated. And it also allows us to retain this signal. And so now we can think about gaining information about cell-to-cell mapping. And so this is just how we make it. I won't go into details, but it's just 8 steps from commercially available fluorescein to get to this final product. This dye, this labeling dye is really selective for hydrogen peroxide. So we do a lot of these types of experiments in the lab once we make our dye, where we expose it or we treat it with a variety of different reactive nitrogen, reactive oxygen species to show its selectivity. And so it's only uncages, or it only gives us a fluorescence response in the presence of hydrogen peroxide. This type of experiments, I take cell lysate and I treat the cell lysate with our fluorophore in increasing concentrations of hydrogen peroxide. And then I perform a gel. So this gel image here essentially is separating all of the proteins in the cell lysate. And what you're seeing by the bands is the fluorescence of the PG1 probe, so the green fluorescence. And so if we don't treat it with hydrogen peroxide, we don't see any labeling. So the probe is not labeling the proteins inside of the cell lysate. And if we take a negative control probe, a fluorophore that does not have this fluoromethylene, again, we do not see signal because there's no labeling. But if we stain this gel to show us the total protein content in the cell lysate, we can see that there is proteins in these bands. They're just not labeled with the fluorescent probe. And so then we can carry this over into live cell studies. And so we can take HeLa cells, which are frequently used in research, stain them with our fluorescent probe in the presence of peroxide and wash the cells, and we see that we retain a signal. So we have this increase in fluorescent signal in the presence of peroxide in our probe. With this negative control probe, we do not see an increase in fluorescence because we're washing out. Again, we're not trapping. This probe can't be trapped in a cell, and so we're losing that signal. And then, of course, without peroxide, we do not see fluorescence increases. We can pick up endogenous changes in peroxide. So we can stimulate peroxide production in some cell lines. So in these raw macrophages, we can treat them with a specific molecule to stimulate peroxide production, and we see an increase in the green fluorescence. But if we treat it with an inhibitor of the protein, NADPH oxidase, the protein that makes hydrogen peroxide, we see a loss of fluorescence. Or if we treat it with an antioxidant, so we're still generating peroxide, but then we're instantly trapping that peroxide with some sort of antioxidant. Again, we lose our signal. So this type of experiment shows us that the signal is coming from hydrogen peroxide itself. And so in this type of experiment, so this is the experiment that really shows us that we're capturing transcellular cell-to-cell signaling between 2 different cell types. So microglia activation is a hallmark, or overactivation, overstimulation is a hallmark of neurodegenerative disorders like Alzheimer's and Parkinson's disease. But how this overstimulation of microglia leads to neuron degeneration isn't really well understood. And so there's one hypothesis out there that says that overly ROS production, increased ROS production by stimulated microglia disrupts the surrounding redox homeostasis, of the surrounding neurons. And so that influx of ROS ultimately leads to their degeneration. And so we can perform this type of experiment where we can take monocultures. So monocultures just means you have one cell type presence in your culture, in your cell culture. We can treat it with our green fluorescent probe and then stimulate hydrogen peroxide production. So in this case, in the monoculture, microglia incubated with lipopolysaccharide or interferon gamma will show an increase in ROS, reactive oxygen species, because this stimulates their production in microglia, but this does absolutely nothing in neurons. So LPS treatment does nothing in neurons, so we should not see an increase in fluorescence. But if we co-culture these cells, so we culture them together, and then we treat them, we should see increase in fluorescence intensity in both cells if there is signaling between one cell type to another, right? And so that's exactly what we see. So we can take the neuron microglia control. There's no increase in fluorescence without LPS stimulation, but if we add in LPS, we get this increase showing us that there's transcellular signaling. And in the monocultures, we do not see an increase. So these are the neuron monocultures that are treated with lipopolysaccharide. We do not get an increase in the fluorescence intensity. So this gives us strong indication or strong evidence for transcellular signaling between microglia and neurons. And we can do an additional control where we knock out the protein that makes hydrogen peroxide in the presence of LPS and microglia. And so if we knock out that protein, we should not see fluorescence. And indeed, that's what we see. Again, giving us additional evidence that it is hydrogen peroxide we're seeing, and that hydrogen peroxide mediates signaling processes between cell types. And so what we're doing now is we're making derivatives that target specific organelles in a cell. We're making derivatives of these probes that emit at different wavelengths. So red-shifted fluorophores generally are much more stable. And they also allow us to do in vivo bioimaging because higher wavelength absorption is much deeper penetrating. And so we can now think about using these types of probes in zebrafish models, which we have, and also in mouse models as well. And so, you know, I did state that there is a large need, one, to really find to find triggers that are able to uncage much quicker. And so the reason why that's important is because if the uncaging process takes a minute to get you a high signal intensity, then you're missing out actually on a lot of spatial information as well, because you're reacting at one point in the cell, but then you don't get this increase in fluorescence until you're at like the other side of the cell, right? Because it takes a minute. And so you can imagine that you diffuse quite a long distance in that 1 minute when you're talking about, you know, something as small as a cell. And so we really need one, quicker triggers to really enhance that spatial information that we're able to obtain. That's really important. And we also, you know, and our lab does this a little bit as well, is try to design brighter fluorophores. There's always a need to design fluorophores that are really bright. And so, you know, one way to do that is we actually calculate the HOMO and LUMO levels of these bottom arrow rings in our systems, because it's thought that systems like fluorescein it's thought that the quantum efficiency of fluorescence of these systems like fluorescein can be modulated by this photoinduced electron transfer. And so you can view these two, this top half and this bottom half, as two separate sorts of molecules, if you will, because they're orthogonal. So they sit like this, and so there's no ground state interaction between the two. So modulating either the donating properties of this xanthine ring or the accepting properties of the xanthine ring, so the donating property of this benzoic acid ring, you can modulate, if you modulate the redox properties of either of the two, you can modulate the quantum efficiency or the brightness that you're able to get out. And so some people will calculate the HOMO and LUMO energy levels of this benzoic acid ring and try to identify substituents that will really modulate this HOMO level such that you're increasing the fluorescence much more. And so it turns out here that the more reducing of the benzoic ring you have on the bottom, the more fluorescence that your actual dye is. And so that's, we do that very lightly in our lab, but there's definitely always use for brighter fluorophores and help designing new ones. So I'll end it off there. And so this is the lab I work for. Chris is a great person to work with. And yeah, I mean, thank you guys for listening and for having me. I apologize for the tardiness.
Thank you so much. Steven actually need to like leave the call because he have like another call. But thank you so much. It was like very interesting. So anyone has any questions, feel free to like unmute yourself.
Yeah, a quick question too about the just like the proteins that you use to like trap the fluorophores inside the cell. Like which actually like how big do you guys like normally go for, and like how like, and like what's your criteria for like a good protein to do that, and how do you know like it's not going to interact like inside?
Yeah, that's a good question. Yeah, so you're talking about like for the stuff like this, right?
Yeah.
So there's no specificity for the exact protein we're labeling here.
Okay.
This orthoquinone methide that we generate in this process is just going to label whatever's around it.
Okay.
Without a care in the world, it's just going to label. So this is good in terms of giving us enhanced spatial resolution, because as soon as it comes in contact with this hydrogen peroxide, it's going to label everything around it and kind of be anchored at that point. So there's no selectivity for specific proteins in particular. But again, this brings up the point that, if we can design computationally triggers that are still selective but faster, our resolution, it'll help us a lot, because you can imagine that this uncaging process, if this takes a minute, this probe is already, somewhere else by the time it labels the protein. And so it would give us a lot more information. But yeah, it's not specific for anything. It's just whatever's around.
Awesome. Thank you, though.
Yeah. Thanks for the question.
I actually have one question from Steven. So Steven, want to ask, as a grad student, are 2 advisors better than 1?
It's so dependent on the advisors you have. I didn't go into it wanting 2 advisors. It just happened naturally. But the 2 people need to work well together. And it's really difficult, I think, to find two professors that, you know, work really well together, that will allow you to have a lot of freedom, right? So they have to trust you, right? Because if maybe you're not doing enough work for one, because you're doing like, you're trying to catch up and do stuff for the other one, then one of them has to be okay, right? And understanding of that. And so you need to find people that are patient enough to handle that, which is difficult to do. I loved it. I liked it because they gave me a lot of freedom. I got to learn a lot of different things and different skills. It was nice having two different mentor types. And so I could choose which one to go to if I wanted a specific, you know, type of mentorship. If I'm saying that correctly. So that was nice. But it's not something I would like, I wouldn't advise people to go into grad school saying they want two mentors or to actively look for it. I would just say let it happen naturally, because it's not common. And it is stressful because it's two group meetings a week. It's double everything. Everything doubles. You know, it's double the things you have to look at for other people in your group, double the, you know, because people want you to look at their papers, their fellowship applications, their, you know, so yeah, you got to know what you're in for. If you want double of everything, then, you know, do it. But I wouldn't recommend it or actively advocate for it.
Thank you. Any more questions?
Yeah, if you go back like the one slide with the little brain, the little neuron. Yeah, that one. I was really interested in like what you said here. So like, do you guys tag like where the stars are at, or like how does this like work like here?
Yeah. So here by the stars, I'm indicating that the probe is fluorescent and turned on. But what you get at is a good point. You know, because in our cell culture, you know, how do we know that we're looking at microglia versus neurons? You know, because I didn't include it here. And so, you know, we have our probe that becomes fluorescent only if it comes into contact with hydrogen peroxide. But, you know, we also need to tag the neuron and the microglia itself. And so there's different antibody fluorophores. So wow, this gets back to like slide, you know, one of the first slides of why it's important to modify proteins and antibodies with fluorescent probes. Because what we're doing here in this top image is we're using an antibody that has a red fluorescent group on it to tag the neuron. So the antibody is selective for neurons. And so that's how we know that what we're looking at here is actually a neuron. We stained the microglia with a blue marker, but I'm not showing that here for the sake of simplicity, but we did stain it with a different marker. So, you know, in our cell culture, we had a green fluorescence, blue fluorescence, and red fluorescence, but all those different colors told us different things, right? Yeah.
Thank you. I really like the colors and everything. That's why I was like really interested here.
Yeah, no, I mean, that's why I got into it. Like, you know, it's simple, right? Like, I have my PhD and everyone's always like, oh, I want to learn this advanced technique and stuff like that. But really, I wanted to make pretty pictures,
That's why I said to Steven, like I really like making pretty plots, so here I am.
Yeah, I want to make pretty. It's a lot of work. I didn't know how much work it was a lot of work.
Yeah,
But you come out, you're making this nice looking data, and learning a lot in the process, which is important. Yeah.
Thank you though.
Yeah, no problem. So everyone's gonna apply to Northeastern, right? For grad school.
Well, I think that we don't have any more questions. So thank you so much, Professor Marco. It was a very interesting presentation.
Yeah, thank you guys for having me. I appreciate it. Thank you for the invite. And if you guys have any questions or anything, please reach out. I'm happy to reply.
Thank you. Bye bye.
Yep. See ya.
Thank you. Have a good one.